Our speaker tonight is Dr. Scott Peterson. I'm pretty excited that he could come because he is connected with the Ventnor Institute. And for those of you who don't know the name, you need to do some research on it because it has been a very active organization right here in the Rockville area. And I've always been curious what goes on behind those doors. So Dr. Peterson is here tonight. We will be glad to have him not only speak with us, but I think he's willing to take questions afterwards. And so it's all yours, Dr. Peterson.
I'm very pleased to be here, and the talk that I put together is really aimed at sort of stimulating thoughts and discussion. So I'm happy to be interrupted, and I'm more than happy to engage in discussion afterwards, and I hope the presentation makes you want to ask some questions. So by sort of definition, I'm an infectious disease researcher, and I happen to study the genomes or the chromosomes of infectious agents. And I became interested in the Human Microbiome project as it started to become defined, and I knew I needed to study that. But I at least the outset, believed that these were two very different fields, and that I had some trepidation about whether I should really be leaving what it is that I was doing for a decade and a half now to study the human microbiome. But I've come to appreciate that, in fact, they're just two sides of the same coin. And so we all sort of, by training as children and throughout life, come to view bacteria as germs and things that cause disease. And in fact, the picture and what we're understanding about the human microbiome, those organisms that live on our bodies and within our bodies, paints an extraordinarily different viewpoint. And that's one of the things I'd like to get across tonight. So 99.9% of the bacteria living within us are non-pathogenic and provide services to their host. So these organisms, and they number in fantastical numbers, that have evolved with us since humankind has existed, have evolved in such a way as to be cooperative. It's called a mutualistic relationship because we derive benefits from those bacteria and they derive benefits from us. And so it's not a parasitic or a pathogenic process, but rather a mutualistic process that we live in harmony. There are exceptions, and this is what ecology tries to understand and study. Every now and then, in a massive population of organisms, one way to achieve fitness in a population where competition is at a norm, and threats and hostility from the human immune system are a norm, is that you'll find cheaters in the population. And the cheaters have evolved ways of living in harmony when that's convenient and improves their fitness, but also have the capacity to take opportunities to infect the human host. And those are the very, very small fraction of organisms that you've all read about in the news. Tuberculosis and Staph aureus and so on. These are the infectious agents, but there's such a tiny, tiny fraction of the species that live in harmony with us. And so it's important to understand and appreciate that balance. So I made reference to Staph aureus, and one thing that I can say about that is if each of you looks to your left and looks to your right, then either you yourself or one of the people you just looked at is colonized by Staph aureus. And this has been true throughout your lifetimes, more or less, and probably very few of you have ever had a Staph aureus infection. And yet, those people who do have Staph aureus infections are likely to face life-threatening illness. So this is an example of how an organism can live in harmony throughout an entire human lifespan, in most cases, but in some situations can become deadly organisms. So I want to sort of turn now to what I consider the premier microbiome. That is the microbiome and the bacteria that occupy our gut. And I largely refer to the contents of the large intestine and colon. So I have this sort of provocative title, The Two-Brained Beast, and it comes from a series of studies and understandings that people have arrived at that have looked at not the microbiome per se, but the actual physiology of the gut. And one of the things that's becoming clear is that the gut has a very conspicuous relationship with the brain, and very likely has a lot of influence over our emotional state, depression, and a variety of other neurological medical problems can be controlled and influenced by the gut. So in the past, when you had an ulcer, if you suffered from a swallowing disorder, had autism or chronic abdominal pain, you were likely to be told by your doctor that you had some psychological issue and that you needed to relax and reduce your stress, or go to see a psychologist to deal with your swallowing problem. And I think over time what we're seeing is that all of these diseases have some component of explanation that has to do directly with the gut. And so you're much more likely in the modern times, and maybe with a more progressive physician, to be told to go see your enterologist, who can help solve some of these issues. So I made reference to the complexity of bacteria that exist in our guts, and the other fascinating thing I learned in preparing for this presentation was that the gut is innervated by 100 million neurons. So the gut is a very long tube, as we know, but it's innervated at a fantastic frequency by the nervous system and in the nerves that connect the brain to the gut. In fact, these numbers are significantly more than the number of neurons that emanate from the spinal cord itself. So throughout history, I think physicians and researchers have always believed that it's the brain that is telling the gut what to do. When you're hungry, you need to eat, and you need to have your digestive system working, and so on. And so the relationship was that the brain was telling the gut, and it did so through these neural connections. But in fact, what I think we're starting to see now is that it's the other way around. And so when you look at the directions of signals that propagate from these 100 million neurons, 90% of those signals are going from the gut to the brain, not the other way around. And so this is an interesting concept in the sense that it's putting an importance on the gut and how much evolution has created a dependency of the gut functioning properly to signal the brain as to how to behave and act. And so again, this goes back to these ideas of autism and complex diseases having an influence that can be profoundly affected by the gut and its contents. So with respect to autism, I want to put this in perspective because it is a relatively common disorder, and many of you may even know people that have family members with autism. But what we're starting to see is some interesting relationships, and I don't want to overstate the relationships, but it's important to understand that when children with autism are given probiotics, for example, they oftentimes see alleviation of their symptomologies. But they're not cured. It's not the answer to their problems. They also, no doubt, have neurological issues that are genetic. But the fact that you can influence the severity of the autism by influencing the microbiome is an interesting concept. In a fairly recent, very small clinical study, 12 children with autism were given antibiotic and 12 placebo controls. And what was interesting is, again, with the antibiotic treatment, they're influencing the gut flora with that treatment, and it impacted the autistic children in a positive way, alleviating their symptoms significantly compared to the controls. So again, these relationships and associations are very interesting and also very preliminary. So this is sort of just the punchline to the gut and the brain, the two-brained beast, is perhaps we think of the brain as being the master, and of course it is in so many ways. But the gut, in many ways, is telling the brain much more than the brain is telling the gut. And so perhaps we need to pay more attention to the gut in our diets and the way that our gut flora impacts our life in various ways, because it's a broad sweeping spectrum of human conditions that will be influenced by the health of your gut. So two little factoids that go along with the signaling potential that occurs in the gut that influences the brain is that the finding that virtually all neurochemicals, neuropeptides that signal in the brain and are thought to promote brain function have all been found in the gut. Nobody quite understands why or how they get there, but they're there. And whether they're being produced by microbes or whether they're being produced by the human brain itself and somehow end up in the gut, again, there's a connection and a relationship. Why are they there? What's their significance? If you look at the gut in terms of the various chemical compounds that are there, enkephalins and also benzodiazepines are two prominent compounds that can be isolated from the gut. Now, in these two cases, they're most likely being made by bacterial species within the gut. But these are powerful opiates and anti-anxiety drugs that have profound influence on brain chemistry and behavior. And again, we find this connection with that and its contents. So I asked this question, can the long brain, and by that I mean the gut, can it learn? And it's an interesting question, and it allows me to tell this story that I recently read of a medical sergeant in the Army who was treating a group of soldiers that were paraplegics. And one of the primary issues that you face as a clinician in these situations is that the soldiers will become constipated and have trouble extracting their colons. And so, as a sergeant in the military, he believed in doing everything on schedule and on a specific regimen. And so, every morning at 10 o'clock, these soldiers were given enemas, and that's how they were able to relieve their constipation. So, at some point in time, after following this regime for months, that sergeant was given another assignment and replaced by some other clinician. And this clinician had a different way of doing things, but strangely, when he went to go and provide his regime to these soldiers, found that all of them were emptying their bowels at 10 a.m. every single day. So in that regard, the gut was able to learn, and it has a signaling mechanism that influences the outcomes of a problem like that. So let me now really formally switch to describing the microbiome. And as I've said, the microbiome is the nature of the organisms that reside on our human bodies and within our human bodies. The gut, the nasal passage, the nasopharynx, the oral cavity is an enormously complex and rich environment for bacteria. Our skin is completely covered with bacteria. And the urogenital system in human vagina. All of these are being studied under this Human Microbiome Project, which is an NIH initiative and a global initiative worldwide. Some interesting aspects of what we understand about this project is that we can think of human beings as being metagenomic or meta-organisms, where we like to always think of ourselves as human and nothing other than human. But we, like so many other organisms on this planet, are actually in the same mutualistic relationship with bacteria. And so we might more properly think of ourselves as being meta-organisms, comprised of both microbes as well as our own flesh and bones. 10 trillion organisms are thought to exist in and around our body. I am not able to conceive of that number, but you can get an appreciation of just how many bacteria that is and what a profound influence their biochemical capabilities would have on our bodies. Everything that they make or break or digest or produce influences our physiology in a very profound way, simply by the fact that they're in such gigantic number. We can't write off them just because we can't see them. The majority of the microbiome is completely uncharacterized, and so our work is cut out for us if we're to really understand how this entity of the microbiome influences health and disease. We have an enormous amount of work to do. The technologies of DNA sequencing and others are paving the way for us to get deeper and deeper into our understanding of these complex ecosystems, but we have an enormous amount of work in front of us. I've already more or less made this point, I think. One other aspect that's particularly challenging when trying to study the relationship of the microbiome to health and disease is the fact that the microbiome is so dynamic and different in every human subject. So it becomes very difficult to point to the individual species that compare a healthy to a diseased individual with any sort of statistical power. There's so much variation. If you take and characterize the bacterial species that are on this part of my forearm and compare it to the ones on the opposite forearm on my same person, there will be very, very strong differences. The same is true for any 3 or 4 inches that I move along the skin surface. So this poses a very significant challenge, but also gives you a sense of the extraordinary diversity of organisms that live within the microbiome. So many of you may not know, but when we're first born, we're essentially sterile. The gut contents are completely sterile. Our oral cavities are sterile. Our skins are not sterile, but are largely populated by, depending on whether the delivery was through C-section or vaginal delivery, will be populated by different types of microbes on the skin surface. But all the development of the microbiome happens after we're born for the most part. It's known very strongly that most microbiomes, for sure the oral and gut microbiome, are strongly influenced by the maternal input. So my microbiome is much more similar to my mother's than it is to my father's, and more related to my father and mother than it would be to a different person, or my neighbor, or a cousin, or an unrelated individual. Let me move forward just in the interest of time. So I'm going to just quickly try to go through 3 or 4 projects that I'm actually involved in at the J. Craig Venter Institute. The first project I became interested was related to dental caries, or what we know as cavities. And one of the things that really attracted me to this particular research area was the simplicity of at least one aspect of the disease. Is the relationship between the microbiome, or the bacteria that are on our teeth, and how that relates to the disease itself. As a child, I grew up thinking that the bacteria that existed on my teeth, I understood, had some relationship to developing cavities, but I didn't understand how. I thought maybe that they were just eating away at my teeth, and that's how the cavities formed. What I now understand, not actually too many years ago, is that it's the acid produced by the bacteria as they eat. So every bacteria likes to eat sugar, and when they eat sugar, they produce acids as a byproduct. And the acids on the tooth surface is what actually eats away at the enamel surface of your tooth and creates a cavity. As the cavity forms, the bacteria can fester and become worse and worse, and the cavity will grow exponentially. So going back to why I got attracted to this particular disease model, it's because there was that clear of a relationship that we have a disease that's caused by acid. It's a simple disease, it's not a life-threatening illness, but it's simple to understand that relationship. And so how do the bacteria influence that?
Quick question. We're always taught that fluoride in toothpaste. Now, does the fluoride strengthen the enamel against the acid, or does it kill the bacteria?
The fluoride is antibacterial.
Cool.
Yeah. So let me show you something really quick that I find mind-blowing. So the basic thing here is that a scientist is taking a pH measurement on the tooth surface and sensing the pH as glucose or a meal is being introduced into the subject. And so what we know is that a healthy person without any cavities, the pH of their tooth surface is about neutral. And right after you eat, within minutes, 2 or 3 minutes, the pH of your dental plaque will drop to just about 6. And then over the course of about an hour after your meal, will return to that baseline pH level, neutral. Now, if you don't have such good oral hygiene or dental care, you have, let's say, small cavities or larger cavities or several cavities, as these kids have. You have a very different situation. Your baseline pH is reduced, In an extreme case, it's reduced significantly to even below this red line. Now, what the significance of this red line at 5.5 pH is that it's at this pH and below that the enamel erosion will occur. Above that pH, the enamel is resistant to the acid. And so it's any time that you spend after a meal below this surface that you're doing damage to your tooth surface. And so the caries activity is propagating organisms in the cavity that then can cause greater degrees of cavity formation through this effect of pH reduction. And so this is why your parents tell you to brush your teeth after your meals and why to brush your teeth 3 times a day. It's to avoid ever being in this situation. But again, the issue here was just to illustrate the relationship between a signal being produced by bacteria and its impact on human health, a very simple relationship. And the questions are, can we understand the bacterial basis for those relationships? Not all the bacteria in the mouth, most of them, in fact, are, again, helping us to stave off those cavity-causing bacteria. And so we want to understand that relationship. How does one give rise to the other? How does the battle get lost? Yes?
Recently, I read that there is concern or suggestion that too vigorous brushing immediately after eating is also damaging. Have some information on that?
I would have to know a little bit more about what specifically they meant. I could imagine it could be more related to gum damage, and by damaging your gums, you can give opportunities for infections in the gum lining, which are much more dangerous. Yes. It does, and that's...
Can you repeat the question?
The question was whether bicarbonate was a good antimicrobial measure, and in fact, it's exactly why, again, it's sort of a household remedy, but one that makes a terrific amount of sense. And there have been clinical trials of other agents that are intended to have that same purpose of buffering pH. But yes, it's absolutely a sound idea. Now, if you have healthy teeth, you don't have really anything to worry about, though, right? So it's in that situation where you haven't been to the dentist in a couple of years. Now you're rolling the dice a little bit. Another study that relates to the gut is related to human obesity. And to do these studies, we're making use of a mouse model that's developed by my collaborator at New York University. Now, one of the things that I like about this particular story is that it's based on an old agricultural practice that has gone on for 50 years or more now. And that in the old times, what's shown here is that antibiotics were given to livestock as a way of fattening them up. And of course, as a buyer, you're not looking to buy a fatter livestock. You want one with lean muscle. But as a trick, because they're selling them based on their weight, these farmers would use this duplicitous trick of administering antibiotics just to fatten the cows up. And so it was based on this observation that my collaborator started to do something he called STAT, which was subtherapeutic antibiotic therapy. So not the kind of doses you would give to get rid of an infection, but doses of antibiotic below that, certainly perturbing the gut flora with this treatment. And so the basis is when he gave this subtherapeutic antibiotic treatment, these mice would develop fat contents 25% greater than the control mice that were untreated. And so this becomes a nice model to make a discrete change to the flora and compare before and after treatment, what's the difference in the microbiome? And how does that relate to obesity? Can we gain insights about what's going on that would create that kind of balance shift? Some preliminary work that's been published by others in looking at human obesity suggests that the change in the microflora influences the way that calories are being used by the human species. And so if we have a set of organisms in our gut that take up a large amount of the calories that we consume, they in a sense help to keep us lean. whereas other organisms are actually able to take the caloric intake we take in and turn it into energy that creates a harder time for us to utilize the calories that we take in. So there's this relationship in the dynamic between how the gut might influence our propensity for adiposity and obesity. Another study that I'm engaged in with researchers at Johns Hopkins University relate to some observations on a particular bacteria that's common in the gut of all humans. So this particular bacteria is called Bacteroides fragilis. And as I said, virtually all of us have this bacteria in our gut. Now there's a particular subtype of that organism that's considered a healthy organism for our gut. It's even one that might be in a probiotic formulation that you could find on the shelves. Encodes a toxin. And now this is not a friendly guy. And what they've shown very convincingly is that when you populate a mouse colon with this particular strain of Bacteroides fragilis that makes a toxin, it creates a chronic inflammatory response in the gut. So by chronic, we mean that there's a week after week inflammation, and that these inflammations predispose the mice and humans to a series of other much more difficult ailments, like ulcerative colitis, like Crohn's disease, and like colorectal cancer. And so these relationships, again, are being pursued by us in this particular study, where we're trying to assess the relationships. And so far what we know are two interesting things. If one examines the flora that's associated with a tumor, a colon tumor, and compares it to the healthy flanking tissue in a colon, the bacteria that are adherent to the tumor are distinct and different than the population that's associated with the healthy colon just 2 centimeters away. The other thing that we see is that there's as dramatic a difference between a colon tumor that arises on the left in your descending colon compared to a tumor that's found on the right in the ascending colon. So again, we have an enormous complexity, no doubt, at play, but we're starting to be able to make linkages in how the relationship between the colon and the gut microflora influence diseases that we've never previously attributed to bacteria. I think last, I'm going to just describe quickly a study that I've just not initiated, but written a proposal to get support to initiate, which has to do with juvenile type 1 diabetes. And just as a reminder, type 1 diabetes is characterized by the complete destruction of islet cells in the pancreas. And so people with type 1 diabetes are essentially unable to make insulin and regulate blood sugar levels. So for years and years, people have tried to understand the genetic basis, and clearly there is a strong human genetic basis for type 1 diabetes. But there's a significant environmental component that's never been understood as to explain why type 1 diabetes arises. Now again, just like in the story I told about autism, there's been some interesting relationships made through the use of antibiotic treatment, as well as various mutant mice models that influence their ability to perceive bacteria in the gut flora. And in both cases, what it suggests is that the gut flora is an influencer of the development and the exacerbated condition of type 1 diabetes. And so many have speculated that the autoimmune associated with type 1 diabetes has to do with bacteria's ability to breach the epithelial layer of the gut and infiltrate into the human system. And through some mechanism that's not understood, the immune system recognizes that invasion as a self-autoimmune activation. And so we're in very early days, but it's a very exciting possibility that there could be a microbial influence, because then the possibility exists that there would be a therapeutic development. So I wanted to wind down by telling one interesting story that I think is phenomenal and has fantastic implications. Recently in the New York Times, maybe some of you read this story also, a woman was in the hospital with an acute Clostridium difficile infection, and she had gone from a 130-pound woman to a 60-pound woman over the course of a few weeks, and was a day or two from death. Her vital signs were crashing, and she was in very bad shape. So, in desperation, her doctor decided to do something radical, which was to provide her with the microbiome, the gut microbiome, via the stool of her husband. And so, using endoscopic procedures, they introduced his gut flora into her. And within 24 hours, this woman was healthy and awake and alive. And within 3 or 4 days, she was eating and back to normal. It was a phenomenal outcome. Since then, that initial study, an additional 13 cases of this therapy has been attempted. All 13 were miraculously successful. So this is just an example, a true life example, of treating a very deadly disease with a microbiome transplantation. So what I ask and what I speculate is that maybe fecal transplantation could be used in the future for treating.
You have to be married to get.
That's why I was saying, if you're more comfortable, you can use your own. But in all seriousness, it would not surprise me that 25 years from now, parents are coached by their physicians to take healthy baby's stool and save it in some medically reasonable fashion so that it could be used for a therapeutic intervention in the future. I truly believe this could be the case. So my final slide, eat right, live well, and then brush your teeth.
Do you use Dial soap?
I actually don't use soap. I don't use detergent soap. I was asked if I use Dial soap, which is a bit personal. I use non-detergent soaps, and I do so, I suppose, because I like the idea of maintaining my healthy flora. Yes.
I was wondering if baking soda is good for the teeth and helps get rid of the acid. Does that be true too? That helps the flora?
Yeah, it's an interesting question. I know of several, let's call them Eastern solutions to medical problems that involve this sort of notion. And somewhat contrary to common sense, though, what's interesting is I drink a drink called kombucha, which is it's a vinegary type beverage that is a ferment, and it has probiotic qualities. But the acidity of it actually helps to alkalinize your bloodstream, and so by ingesting acid, sometimes you're able to influence an alkaline response. So it's a little bit counter, but your concept is actually correct, and we do have a lot of evidence to suggest that a variety of colon and stomach cancers are directly influenced by too much acidity. And too much acidity in the bloodstream is a sign of poor health and a high risk indicator for various human diseases. So, yes.
When our children were little, we had a family physician, Dr. Gude, along with his, who never prescribed antibiotics or anything unless he also prescribed some sort of food that would have bacteria. It's a yolk, natural yolk.
How many years ago was that?
Pardon?
How many years ago was this?
Well, our oldest child is 42.
Wow. Very progressive doctor.
Yes, it was. And you know what? We never had any problems that other people had when they took antibiotics. We didn't have to take it off until we did.
Yeah.
We didn't have trouble.
So, I need to be careful not to get going about antibiotics. let me be very clear that they save lives, and I don't recommend anybody to take lightly the idea of treating infectious disease with antibiotics. But we all know that they're badly abused and badly overprescribed for viral illness and a variety of other things. Nevertheless, one of the things that I do, and as I said, I study infectious disease, and one of the things that we've come to learn is that so many of the bacteria that cause infections have mechanisms to hide, some through invading human cells. Even if that's not the primary mode of the infection, enough of the population is able to do that. And they're able to essentially hide from the antibiotic challenge. So what they end up getting exposed to is a little tickle of antibiotic. And what that little bit of antibiotic does is induces mutations in the organism that cause resistance to the drug. And so by treating with an antibiotic, you're actually, even if you follow the regime, you suffer a real consequence of potentially selecting for antibiotic-resistant organisms. So with organisms that are involved with recurrent infections like Staph aureus, if somebody has a skin abscess, the first thing your doctor will do is look for another, because it's a recurrent phenomenon. And so you treat that skin abscess, and if it comes back 6 months later, you can almost guarantee that it will be drug-resistant to whatever you were treated with. So it's a double-edged sword, is I guess my point. We need to respect antibiotics. Yes, sir.
Sort of related before a prostate biopsy and for an antibiotic.
Probably very smart.
Messes up.
Yeah.
What you're talking about.
Yeah. Well, doctors have a very good reason for doing that, because anytime you're having a medical procedure, especially one that involves contact with the bloodstream, you have a very high risk of even naturally occurring flora from your mouth, for example, entering your bloodstream and causing endocarditis, which is deadly. So it's one of those cases where the frequencies of that occurrence may be very low, but when you're the victim, you want your doctor to give you antibiotics, and it's well advised. Yes, ma'am. So the basic gist was, again, the notion of antibiotics being a double-edged sword, and the idea that it's very commonplace for people on antibiotics to get overwhelmed by yeast infection. This is a particular problem for women in their urogenital system. Recurrent UTIs are a major problem for mainly women, but also men in this country. So you're right, and I think again a good physician will be a little bit ahead of the game and prescribe countermeasures like probiotics, like cranberry juice, like things that will help to maintain that balance and not allow the yeast to overwhelm, because it is almost predictable, especially for some women. It's a guarantee. I'd love to talk about both those things. The first question was related to the reduced use of fluoride. Two things I could say. One is, working in developing countries and studying dental caries, it's absolutely true that people without fluoride have much worse dental problems. Part of that is reduced access to dental care, but the fluoride absolutely has positive influence on caries development. There is a downside to fluoride, as it is being implicated more and more in terms of a variety of negative health impacts, and I'm not probably expert enough to comment specifically on which ones I believe are valid. But for sure there is this tendency to want to reduce our dependency on fluoride for oral health. Countries like Denmark and Scandinavian countries that are small and wealthy have taken the approach of just eradicating caries from their population through fluoride treatments that are given at the dentist's office, so liquid fluoride treatments, and vigorous and routine dental checkups. They've literally eradicated dental caries from their country. So there's several ways to win the war, but some have pros and cons depending on your economic affluence, I suppose. Yes. Sorry, can I stop you? I wanted very much to answer your other question. The second question she asked was about cleansing, and I don't know if any of you have tried to do a colon cleanse in your lifetime. I have, and I did it purely as a scientist. I was curious to know what would be the impact. My wife, on the other hand, is a big believer in this, but I just, literally as a human guinea pig, I wanted to see. And so I've tried 3 or 4 different types, and some of them are better than others, but I would say that it had a profound effect. Energy level, and a secret for people who care is my skin was never more healthy or vibrant or young looking as after I did that cleanse. But I think again that this is an important concept, and whether a cleanse is the way to establish gut health, I think it certainly gets in that direction. Whether it's sufficient to maintain and truly have a healthy gut, I don't know that I can answer that. But I am a believer. Sorry. No, a colon cleanse is usually an herbal remedy that's taken throughout a 1 week or 1 month course. I'll come back to you. How about that?
Are the bacteria that are associated with dysentery the same bacteria that are the oral bacteria that are being implicated in arterial inflammation?
Often.
And how is it actually gum damage and how that's true? So this is
Exactly.
This is a commensal bacteria that's basically in the mouth that can be a real damage.
Yes.
What's the question?
So the question was what the relationship is between the oral cavity and the microbes that are there, and cardiovascular disease, and as I already mentioned, endocarditis. And there's an absolute relationship, and it almost always involves physical contact of the bacteria in your oral cavity with the bloodstream. And so through the bloodstream they can migrate, and they like to end up in the heart valves. They're particularly likely to end up in artificial joints and prosthetics. They just have a propensity to find their way there and form a biofilm, which is a three-dimensional structure of growth. And the immune system doesn't know how to deal with it. And so they can exist there for years and years and years. It's a very, very common problem with joint... Yes. Now, endocarditis will kill you long before that. Yes. But these kind of chronic infections involving artificial joints can go on for years, and it's very debilitating. Let me go back here and...
Well, Steve would say that the various gut cleansing techniques are useful, various herbal remedies. Is that correct?
That is what I said. That's the million-dollar question. I've often, I pay attention to the cosmetic industry and GNC, and I think it's all nonsense for the most part. Now, certain things work, and so I like to have an objective viewpoint about what works, and then I want to know why. But the cosmetic industry wants to sell you everything and tell you why, but it's probably not the case. But with the gut, we just don't know. Is it just simply that you're promoting healthy gut bacteria? I know one of the things that you benefit from in any cleanse, and I also sometimes will drink vegetable juice, what's called juicing. And one of the things that's for sure the case about that is that by doing that process of taking the pulp out of the vegetables, your body has an enormous ease of absorbing all the nutrients from that dietary intake. And as a result, your body doesn't have to use so much energy to digest the steak that you ate in 3 minutes. And so you end up feeling very vibrant. So that may not have anything to do with the microbial flora. It may just have to do with gut energetics. I don't know. But I think it's a combination of all these things, that the cleanse you're eating easily digestible, easily absorbable foods while you're doing a cleanse generally, and the herbs themselves probably do influence the gut flora. But somebody should do the experiment. Then you could sell this stupid stuff and make a lot of money, right? Show how it works, and I'll buy it. Answer would be probably not the same organism. Sorry, the question had to do with whether there were relationships among the bacteria that cause calcification of teeth and tumors and other kinds of human disease. And I think that the answer is unknown. But my guess as to what the answer is that they're most likely caused by independent bacteria, all with that common outcome. Whether they're actually the ones making the calcium phosphate or whatever, I don't know. But through some collaboration, they're causing the problem. Yeah. Bacteria can get pretty much everywhere, and how successful they are once they get there, it has a lot to do with the human immune system. Remarkably efficient beast, because we're constantly being invaded, and we don't get sick. We should get sick a lot. Yeah, good question. So more and more you find doctors prescribing probiotics or yogurt or things that will help maintain your flora during an antibiotic treatment. And so that's essentially what seems to be taking hold. Now the probiotic industry is another one that I have a little skepticism. You eat yogurt, I know there's some probiotic quality to eating yogurt. Some of the formulations that are you see more and more and more, and that makes me suspicious. Does everybody really know what's going on with those probiotics, or is it sort of happenstance just like yogurt? Yeah, if you eat this, it sort of makes but nobody's showing the impacts. Take this probiotic and look how your flora changes. Show us. But this is what people do, and yogurt is a great home remedy for antibiotic treatment. They're certainly affected by the treatment, but it's a transient service that they're providing. Yes, that's a very good question. So to some extent, any time that we eat anything, it has bacteria on it, and the acids in our stomach are essentially what was being asked was, don't they damage or kill those cells? And the answer is absolutely. But when you eat, first it buffers the pH of your stomach, so it's not as low as it's sometimes you read pH of 1 or whatever. During a meal, it will be closer to pH of 4, and there are several bacteria that can tolerate those pHs for short periods of time. I had a second point, but I lost it. Anyway, the other point was when you ingest 10 to the 14th of anything, some are going to help. Yeah. So the question was, if we do a sequencing survey to determine how many species are present in the gut, how many do we see? And the answer to that is this is one of my scientific interests. So the answer that most people would give you is that the gut has on the order of 500 species, and the oral cavity has something on the order of 1,000 or 2,000 species. The issue, though, is that even with our gargantuan capability of sequencing DNA now, whenever you have organisms that are arranged at a very broad range of abundance, when you apply the approach of just sequencing things at random, what you're doing is you're getting the sequence of the surface most abundant organisms. And what those guys below you never observe. And so that's where I just give you that answer as that's what we know now, and I think it's much, much deeper than that. Yes, sir.
How does alcohol affect the flora?
That's a good question, and I don't know that I have an answer. I'm sure somebody's going to study that at some point.
I find it gets better than safer.
Yeah, right. Yes, sir. Yeah. Okay, let me see if I get all those. So, I'm not a physician, so I've been pretending a lot tonight. So I don't know the answer to your first question. Yes. The question was whether fasting for a day every so often was a good idea. My answer to that is if the yogis from India do it, then so will I. They know a lot more about human health than I think we do, or at least they respect it and practice a lifestyle that promotes human health. Your second question is fantastic, and the subject of a grant proposal that I wrote and was not successful in getting funded. But it has exactly to do with the dental caries model. I can give the answer. So we've been trying to make associations of which bacteria are associated with kids with caries. And so we have a couple of culprits that we've identified. And so we start following these organisms, and what we see is that we follow these children longitudinally. So at the beginning of the study, they're caries-free, all their teeth surface. And at some point during the study, they'll develop a cavity because they don't have fluoride in their water. These are Brazilian children. And so we have the opportunity to ask, among the culprits that we've identified, do their abundance change as a kid develops caries? And sure enough, they do. But we came across a couple of samples in our small study at this point where the levels of those bacteria that are associated with dental caries elevated, and the kid was clinically caries-free still. So do we have a forecast? Do we have a system that we can see things before they're going to clinically manifest? That's a billion-dollar idea. So I'm so anxious to understand that on some problem that's more significant than dental caries. Does the tumor-associated bacteria that I described tell a story before the tumor gets there? Can we sample the gut mucosa and see collections of bacteria that are causing inflammation and going to give rise to a tumor if we don't intervene? That's a very important idea. Yeah, so I don't have a great answer. I think there's a lot of anecdotal information. One very interesting scientific project that I'm aware of, sponsored by the NIH, is a project called the Welderly Project. And so what they've done is they've looked for people 90 and 100 years old that are in perfect health. They've never had health problems in their whole lifetime. So much of our analysis of people's genomes and chromosomes and mutations are looking at sick people with disease. And this is a project geared towards understanding what are the genetic traits that make these people healthy. It's a fantastic idea. So simple in concept, and yet it's the first time I've ever seen something like this being done on a good scale. So I think we'll maybe in 5 or 10 years have an answer to your question. Sure. I was ignoring somebody. Yes, ma'am.
Question about bunnies who routinely eat their nighttime droppings.
I'm out of my domain, I think.
What?
I'm out of my strength now. I don't know. Okay. And so is there maybe some advantage to that the rabbit benefits from? And I don't know the answer to that, but it would make an interesting scientific inquiry, I suppose. Mm-hmm. Much crazier things are being done. I think my answer to that is the question is whether there might be an advantage to going back to taking antibiotics intravenously as opposed to through oral route. I think the only time where that becomes important is in that case that I described where you have an infection and you're infected with an organism that has a propensity to know how to hide. So Borrelia or Lyme's disease is a very good example that we probably all know about. In my very strong opinion, the antibiotic regimen for treating Lyme's disease is inappropriate. And I say that because if you're lucky enough to have become infected by Borrelia, by a tick, and you immediately go to the doctor and get oral antibiotics, the 30-day treatment that's standard is adequate to alleviate that infection. But there are exceptions to that, and there are other cases where people don't get treatment soon enough, and yet the treatment is the same. Now, if you've gone 1 week beyond the tick bite, or 2 weeks beyond the tick bite, or longer, you just have an increased probability that that bacteria has wound up somewhere where it's impervious to the antibiotic treatment. And this is the chronic disease that you hear people talk about. I don't have any energy, I can't think, I'm going to lose my job. It's tragic. And it's because they can't readjust the way they treat the illness. It's perfectly appropriate to give 30-day antibiotic regime, and it absolutely eradicates infection in 90% of the cases. But those other 10% that's not so lucky have a nightmare for the rest of their lives because they didn't get treated properly early. It's a shame. I think getting back to your question, the IV antibiotic route is absolutely appropriate in these cases to make up for the fact that you didn't catch it early. You need to be super aggressive to eradicate the infection. You can't leave the stone unturned. Yes, sir. At the Institute, we've done that on a number of occasions. It's not my field of strength or knowledge to... Sorry, the question was whether we sequence organisms known as archaea. It's been thought for many, many years that there were two types of life on the planet, one being referred to as prokaryotes and the other being referred to as eukaryotes. Eukaryotes have nuclei and represent anything from a microbe like yeast all the way to human beings. We're all eukaryotes. A third taxonomic group, ever bit as prevalent and important as eukaryotes or prokaryotes, was very recently described in the past 15 years or so, named archaea. And it was just a previously understood, poorly studied group of organisms that now have a real place. And yes, we do look at those. They oftentimes are what we call extremophiles, which means they can live at 90 degrees Celsius temperatures or in pH 0 acid mines or you name it. They have some tremendous qualities that one would never imagine on this planet. Sorry? Archaea are in the gut. They're not extremophiles, thankfully. Or maybe that would be a good thing. I don't know. But no, yes, there are archaea in the gut. Yes. Certainly there's a spectrum of single-cell eukaryotic organisms in the microbiome of all parts of our body, and viruses. Thank you.